Hi, I'm Tarina Chen, and I spent this 8-week course researching Menkes disease and its associated gene, ATP7A. Here's a short introduction to what Menkes disease is. The gene that causes it is ATP7A, which codes for the protein ATPase copper transporting alpha protein. Menkes disease occurs when mutation of the ATP7A gene results in a non-functioning ATPase. Symptoms of Menkes disease include characteristic kinky hair, which gives its nickname kinky hair disease, and loss of neurological function development, seizures, dry skin, failure to thrive, and typically results in death for infants around 3 years of age. All of these symptoms are caused by a lack of sufficient copper during infancy. Menkes is also known as kinky hair disease, as mentioned earlier, steely hair disease, and Menkes syndrome. The image to the right shows what kinky hair looks like and its texture in comparison to normal hair. Menkes disease has a history that spans almost a century back, where kinky hair was first linked to copper deficiency in 1937 in studies relating to sheep. Menkes and his team in 1962 were the first to describe the disease's symptoms in humans. As such, the disease was named after him. The term kinky hair disease was coined in 1966, and in 1972, copper deficiency was linked with Menkes disease in humans. Genomics plays a role when studying Menkes disease. A general definition for genomics is the study of molecular biology in regards to the structure, function, changes within, and evolution of part of or the whole genome. As Menkes disease is genetic in nature, genomics helps determine its cause, diagnosis, and evolutionary history. Menkes disease is caused by a mutation, is largely inherited from mothers, which determines it to be mainly caused by genetics and not environmental factors. Comparison of genomes also helps identify other organisms with similar strains of ATPase, which helps with finding model organisms that can be used in clinical trials. Genomics can also help with diagnosing individuals early, which helps with administering treatment as soon as possible. Continuing to study Menkes disease may result in development of better treatment for individuals affected and potential application of modern biotechnology such as gene editing as methods for other versions of treatment as well. In general, learning more about a disease contributes to modern medicine and also gives hope in avoiding the tragedy of losing one's child. I've personally always had interest in incurable diseases and the ethics surrounding them, as treatment, while they may not result in anything, the chronically ill still deserve to be treated. It is a moral conundrum that I would like to see more views on. The gene ATP7A is located on the X chromosome, and deleterious alleles are autosomal recessive. There isn't a single SNP or mutation that leads to Menkes disease. So long as the mutation or mutations result in a non-functional ATPase, the individual with said mutation will have Menkes disease. Larger mutations result in less functioning or non-functioning ATPase, such as frame shifts, deletion of large segments of the genome, and inversions. This is a diagram on the general expression pathway of ATP7A. Regulatory elements such as SN1 and HIF2 alpha bind to the promoter sequence of ATP7A, which increases the amount of expression of the gene. The mRNA goes through pre-translational editing to remove introns, whereas exons are pieced together and have a 5N cap and a poly A tail added on. The mRNA gets translated into ATPase copper transporting alpha protein, which is stored in the Golgi bodies and inserted into the cell membrane when copper levels in a cell need to be regulated. As for a gene sequence, the entire sequence measures out to be 140 kilobases, while its exons measure 8.5 kb. The figure shows a count of ATCGs in exons only. There are portions of these exons that are not translated. There are 23 exons in total, where there are 57 different spliced variants, 222 orthologs, 21 paralogs, and 10 phenotypes. Figure 3 shows that this gene is expressed in most cells in the body, though more so in the bladder, lungs, thyroid, and skin. The protein produced from ATP7A is ATP7A's copper transporting alpha protein, or Menkes protein. This protein is a transmembrane protein responsible for regulating copper levels inside the cell, transporting copper in or out based on homeostatic conditions. The protein functions with the binding of a phosphate group from ATP, where the structure changes to transport one copper ion into or out of the cell. Copper is an important component for proteins, organelle function, and development of multiple body parts, including the brain. So the lack of copper is a severe issue for the body, especially during development. Mutations in the genome result in varied effects on the protein, where loss of function ranges in partial loss or complete lack of function. Partial loss of function is associated with occipital horn syndrome, while total function loss is a trait of Menkes disease. The sequence to the right is the canonical isoform of Menkes protein. UniProt has 6 other sequenced isoforms and 17 potential ones that were digitally sequenced and compared. This here is a 3D render of the Menkes protein. The protein is made of 1,500 amino acids. The Menkes protein is part of a protein family P-type ATPase, which relies on ATP catalyzation to function. This family of proteins is present in eukaryotes and prokaryotes. There are 349,000 proteins in this family that are in 34,848 recorded species. This slide is a figure that compares the Menkes protein of Tibetan sheep with 19 mammalian species, where 2 orthologs of Menkes protein appear in the Bovidae family. Furthest derived from Tibetan sheep is the Menkes protein found in mice. Another protein sequence comparison from the human Menkes protein sequence compares it to the ATP7A and ATP7B strains of rodent and plant species. The most similar species to humans is the Chinese hamster, and the least similar, yet still comparable to the human Menkes protein, is the Oryza sativa japonica group, or the Japanese rice family, and Arabidopsis thaliana, or thale cress. Diagnosis of Menkes disease takes several forms. The most recognized form of diagnosis is via symptoms of Menkes disease, though this comes with drawbacks in regards to treatment, which I will elaborate on later. There are genetic tests as options as well, as mothers can test for being carriers of a defunct ATP7 gene, or infants can have their DNA tested. Another proposed method is in testing for biomarker chemicals or neurochemical makeup. Biomarker chemical tests check for low levels of ceruloplasmin and copper in the blood, while neurochemical makeup tests look for abnormal levels of dopamine, norepinephrine, and dihydroxyphenylglycol. As of now, there is no cure for Menkes disease. It can be treated with lifelong copper histidine injections, but its effectiveness varies based on when Menkes disease is officially diagnosed. Typically, symptom-based diagnosis occurs around 2 to 3 months postpartum, but earlier diagnosis can offset the Menkes disease on development. However, many parents of individuals afflicted do not maintain treatment for a lifetime. While treatment is inexpensive, the disease cannot be cured, and some see treatment as a futile effort to keep the patients alive. Overall research indicates that 1 out of 50,000 children are affected by Menkes disease with regional variation. Data from several Central and Western European countries show 1 out of 298,000 live birth children are affected. In Japan, the rate of Menkes disease occurrence is 1 out of 360,000. Australia has a significant higher occurrence of between 1 out of 50,000 to 1 out of 100,000 children, though there may be other factors at play, such as the founder effect from its original population. As a X-linked disease, Menkes affects male offspring disproportionately, as males only have 1 X chromosome and therefore only 1 copy of ATP7A. While females rarely get Menkes disease, there have been cases of female children with Menkes as well, depending on the proportion of X inactivation of functioning to non-functioning genes. Adults can also develop it later in life, as there have been cases of individuals up to 50 years of age that have Menkes disease. As of now, there are no known sociopolitical implications of Menkes disease, as it is both a rare occurrence and also happens mostly in infants. There are potential undocumented effects on adults, such as Menkes disease occurring with other chronic symptoms of copper deficiency. These chronic health issues aren't protected by the Genetic Information Non-Discrimination Act of 2008. It can also be used to fearmonger and spread misinformation about genetics and genetic diseases due to the main affected party being children. Patients in less developed countries may also not be able to receive constant treatment for Menkes disease as Menkes is lifelong. There are also ethical concerns regarding studies of Menkes disease. For one, because of its frequency to occur in infants, test subjects and studies would mainly be done on children, which is of ethical concern. While some of these treatments can be done on animal models, effectiveness varies on human patients due to species differences. In others, the treatment of chronic diseases. As Menkes has no cure, should individuals with Menkes disease be treated? Currently, the consent of parents is prioritized, as are the emotional and mental states of those surrounding the patients. As such, studies are kept in check by human input. And here are my works cited. There are more in the attached document, and thank you for watching.